Dubey Mishra, a gold medalist PhD in criminology and forensic science. She has qualified UGC NET and was awarded prestigious Junior Research Fellowship by Directorate of Forensic Science Ministry of Home Affairs, Government of India, New Delhi. Dr. Swati Dubey Mishra was awarded Junior Scientist Award in National Seminar. She has actively attended more than 75 national and international conferences, workshops, and seminars. Dr. Swati Dubey Mishra has many research papers in various national, international, and regional journals, conference proceedings. She is a meticulous professional with nearby 40 years of extensive experience in the field of teaching, learning, research, and development in forensic science. Dr. Swati Dubey Mishra is a life member of Indian Science Congress Association, Kolkata, Forensic Science Development Society, Lucknow, Global Initiative Academic Network, Ministry of Human Resource Development, Government of India, IFS, Pune. Winning an award is always an achievement. We hearty congratulations for your incredible success, rewards, and achievements, ma'am. Now, the time is yours.
Thank you so much, ma'am, for introducing me in such a wonderful manner. And sorry for inconvenience. Yesterday, due to weather, I could not join. Now, the topic of my presentation is role of microbes in forensic investigation. So here I want to ask from the participant, is there any idea about the microbial forensics? If yes, then please raise your hand. Yes, no clue, ma'am. Okay, no problem at all. Sharma and Anjali has raised the hand. Yes, you may unmute yourself. So before starting the topic, you all should know what is the exact meaning of forensic science. Forensic science is nothing but it is the application of science to criminal justice system or application of science to aid the law. Okay, so what is the meaning of microbial forensics? The role of microbiology for solving the crime is known as microbial forensics. Yes, sir, you are correct. So here the topic of my presentation is the role of microbes in forensic investigation. As I already told, the application of microbes to solve the crime is known as microbial forensics, or in another word, we can say forensic microbiology is the use of microorganisms to help solve crimes by examining microbial evidences from various samples. Now, here the question is, what is the importance of microbial evidences? You all should know about that, because there are lots of techniques by which we can solve the crime and we can investigate the crime. So why it is the need? Why the need of forensic microbiology? So the main importance is that microbial evidence can provide unique information that is often difficult to obtain through traditional forensic methods like forensic entomology, forensic medicine, etc. So it is an emerging field. Now we have to know about that. What are the main applications of microbial forensics? So you all have know about the post-mortem examination. Okay. So what is the meaning of post-mortem examination? Everybody know? And what are the main objective of post-mortem examination? So there are main three objectives of post-mortem examination. The very first is to identify the time since death, to identify the cause of death, and to identify the manner of death. Now, microbes are used to identify the time since death. In traditional method, we use the insect to identify the time since death, and it is also known as post-mortem interval. So the very first application of microbes to identify the time of death. Now, what is the second application of microbial forensics? There are so many applications, but due to time constraint here, I am sharing only few applications. So second one is tracing the source of biological material. Microbial profile can help trace the origin of material like soil, water, and biological tissues. We will discuss further what are the exact role. Now, next application is bioterrorism investigation, identifying the tracing pathogen used in biological attack. We worried about the corona, and earlier we thought that it is maybe it is the bioterrorism attack from the China. So we can also understand the role of microbes in bioterrorism investigation. Now we can take it one by one. As I already told, that microbes are used in post-mortem to identify the post-mortem interval or time since death. So how can it possible? So there are two steps for identifying the post-mortem interval. First is decomposition process. How microbes are used for the decomposition of dead body. So bacteria and fungi break down organic matter, producing gases and fluid that attracts insect and other organism. And we know very well the cycle of insect: larva, pupa, and then adult. So, in which state we find the body, and in what is the stage of insect on the body, by which we can easily analyze the time since death. So, first is decomposition process, and second one is microbial succession. Specific microbes appears at different stages of decomposition, providing a timeline. I am in Madhya Pradesh, and you are many of you joined from different states of the India. So we know very well there are so many varieties of microorganism present in various places, and they are also the unique features of the specific place. So we can also identify the place of the crime with the help of microbial forensics, isn't it? So this is very, very important clue for identification of postmortem interval. As I already informed you that, or told that, the period between death and the discovery of the body is known as postmortem interval, and we can examine the postmortem interval with the help of microbial analysis. So by examining the types and quantities of microbes present at different stages of decomposition, forensic scientists can estimate the post-mortem interval, or you may say time since death. Now come to the human microbiome in forensic science. You already heard that we can identify the person with the help of their sweat smell, isn't it? Means these are the unique signature of each and every individual. Similarly, microbes are also the individual feature of any of the person. So microbiome is highly individualistic in nature. Here we can say that unique what is the meaning of unique microbial signature? Each person's microbiome is unique, influenced by factors like diet, environment, and genetics. So we can also individualize the person, or we can also identify the person with the help of microbes. So as I already told, each person microbiome is unique, influenced by a factor like diet, environment, and genetics. So microbial fingerprints can be used to identify individual or link them to specific location. Here, I would like to ask one question from the participants. You already heard about the fingerprint. Fingerprints are the unique characters. Now, you already heard about the DNA fingerprinting, because like today's media is very, very fast, and you all see about the some crime investigations, so like CID, etc. So you heard about the DNA fingerprinting. You heard about the fingerprint. You heard about the brain fingerprinting. So why we use always fingerprinting word after the identification technique? Have you any idea? Anyone have any idea?
Ma'am, it is specific to a specific person. That's why we use fingerprinting. It
Yeah,
is
yeah,
an
yeah.
individualistic.
Very good. It is individualistic, but why we use fingerprint word? It is individualistic, like DNA is individual to individual. Similarly, brain waves are also individual. Similarly, we can say microbial for fingerprinting means microbes are also unique for each and every individual. But why we use fingerprint word? Here I want to tell all of you that fingerprinting technique is first authentic technique which is used for identification of the person. So for the honor of this technique, we use fingerprint word after the each and every individualization technique. Understood or not? Isn't it? So for the honor of that technique, because we can also identify the uh, in identical twins with the help of fingerprints, but we cannot identify the identical twins with the help of DNA, isn't it? So till date, no technique is replaced, can replace fingerprint technique. There are so many emerging techniques. There are so many biometric techniques like DNA fingerprinting, brain fingerprinting, eye scanning. In eye scanning, there are two techniques: iris scan and retina scan. But no technique can replace fingerprint technique. There are so many advantages of fingerprint. So that is why, for honor of the technique, we use always fingerprint word for with each and every identification technique. So come to the next point. Here we are discussing about the human microbiome. So as I already told, microbes are also unique for each and every person. So where we can apply these microbes in forensic science. So there are two types of application of a human microbiome. First is skin microbiota. Okay. Analyzing bacteria from skin cells left on the object can help identify who has touched them. For example, here I am touching the mobile and my skin cell is transferred onto the mobile. So with the help of specific bacteria, we can identify the person. As I already told, microbiome is unique to person. It's depend on his or her diet, environment, etc. Now, second one is body fluids. So DNA is present in all nucleated cell. Okay, in blood, saliva, etc. So we can also identify the microbes with the help of some biological fluids. As I already told, DNA is present in nucleated cell like blood, saliva, rooted hair, isn't it? But in blood, we can extract DNA only from WBC. Why not from RBC? Have Have you any idea why we cannot extract DNA from RBC? You all are from science background, that is why I'm asking. And I think each and every lecture should be interactive. That is why I'm always asking the question in my lecture.
Ma'am, RBCs are enucleated cells.
Yes, yes. RBCs are non-nucleated in mammals. Exception is camel.
Camels.
Camel RBCs are nucleated. That is why we always extract DNA from WBC, because human WBCs are nucleated. And lower quadrates have nucleus in RBCs. But in human, there is no nucleus in RBC. So we always use WBC for extraction of DNA. Now, here we already discussed about the human microbe. Now, how soil and environmental microbes are useful in forensic investigation. So, soil microbes can useful to identify the place of the crime, because as I already told, I am from Madhya Pradesh, and the composition of soil in Madhya Pradesh in different and in other states is different. Okay, if body is transferred after committing the crime from one place to another place, so with the help of soil analysis, we can identify the place of crime, because specific microbes are present in specific soil. Okay, so with the help of soil, we can also identify the place of the crime, or you may say geographical location we can identify with the help of microbes, or we can link the suspect and belonging with the help of soil. Now, environmental microbes. You all heard about the diatoms. Okay, diatoms is unicellular alga. So diatoms also has unique characters, and it is very what we can say, it cannot destroy easily because of the presence of silica. Okay, so with the help of diatoms and other microorganism present in the body, water body, we can identify the post-mortem and anti-mortem drowning. You know very well what is the meaning of drowning. Death from drowning. Okay, so someone has dropped the body into the lake after committing the crime, after dying the person, that is known as post-mortem drowning. Or someone die after drowning, that is known as anti-mortem drowning. So we can identify the post-mortem and anti-mortem drowning with the help of microorganism. This is very important application. Now, plant microbes are also very important to for in forensic investigation. How pollen grains and leaves also are unique and vary from place to place. So we can, and the microorganism produced on the plant body are different. So if any suspect has some leaf on his or her clothing, so we can identify the person with the help of that botanical evidences like pollen grains, like leaves, etc. Now, here I want to discuss the forensic investigation process. How forensic investigation is done in case of microbial forensic. So the very first step is crime scene investigation. After filing the FIR, investigation official will go to the crime and investigate the crime. So first step is crime scene investigation. Then after that, securing of crime scene and documentation of crime scene. Securing of crime scene as per the need. If the crime scene is outdoor, so we use barricade. And if the crime scene is indoor or inside the building, then we close the door and we appoint any guard, etc. Okay. So the very first step is secure the crime scene and then documentation of crime scene. There are so many methods of documentation of crime scene. The very first method which we use at crime scene is photography, scaled photography, colored photography, videography, note making, and sketching. These all are the process which we use for documentation of crime scene. And then after, we search the evidences. After searching the evidences, we collect the evidence and transport and storage for laboratory analysis. Isn't it? So in crime scene investigation, the very first we secure the crime scene and then documentation the crime scene and then searching the physical evidences and then collection and preservation of physical evidences for the laboratory analysis. So third step is laboratory analysis. And then what we do after receiving the sample in laboratory, first we prepare the sample. So in case of microbial forensics, we prepare the sample for microbial identification. Sample may be soil. In next step, we will learn in detail. And next step is determination of microbial composition and comparison with reference database. And then forensic interpretation. In forensic interpretation, we can identify the post-mortem interval, we can identify the geolocation, and we can identify and associate the person with the crime. And after this process, we generate the report, and that report is ready for court presentation. We can go through one by one. Crime scene investigation. Samples are collected from the crime scene. As I already told, first secure the crime scene, then documentation the crime scene, then searching the evidences, and then collect and preserve the samples for the laboratory analysis. So samples are collected from the crime scene, including soil, water body fluids like blood, urine, saliva, semen, if sexual assault case, and decomposition remain means if any leaves or pollen grains or any bone or any tissue is found at the crime scene, we collect. And then transport, sample transport and storage. Samples are securely packaged to prevent contamination and stored under controlled condition to preserve microbial integrity. Now, in laboratory analysis, microbes are isolated from the samples. Whatever sample we have collected, we isolate the sample from that. We isolate the microbes with that sample, and then we identify the microbes. So there are so many techniques for identification of microbes, like DNA sequencing. In DNA sequencing, we can use polymerase chain reaction for identifying the microbial species present in the sample. And then microbial community analysis. The composition of microbial community is determined and compared with reference database to provide context and relevance to the investigation. and then forensic interpretation. In that step, we identify the microbes, and after identification of the microbe, we will able to know about, maybe not 100% sure, maybe the post-mortem interval or the microbial, with the help of microbial succession pattern on decomposition remain are analyzed to estimate the time since death. Geolocation means after analyzing or after identification of the microbes, we can identify the, maybe why identify the geolocation means microbial profile from soil or water sample are matched to a specific geographical location. Identification and association, unique microbial fingerprint link individual to object or location, provide crucial evidence. And report generation, after interpreting the data, we will able to generate the report. Findings are compiled into the detailed report, interpreting the result within a legal framework and preparing expert testimony for court. And at last, court presentation, the microbial evidence is presented in court with experts explaining the methodology and finding while defending against cross-examination. So this is the whole process for forensic investigation in forensic microbiology cases. So in forensic microbiology or microbial forensics is one of the branch of forensic science in which we use microbes like bacteria, virus, fungi for identification of crime or for solving the crime. And these all are the evidence. These evidence are corroborative evidence. We have to analyze some other evidences also for identify or for solve the whole crime. So at last, we conclude that microbes play a crucial role in forensic analysis by aiding in the determination of post-mortem interval through the examination of microbial succession on decomposition body. Their unique microbial signature help identify geographical origin and track environmental exposure. Additionally, microbial DNA can link suspect to crime scene or victim to trace evidences. Advances in microbiome research and sequences technology have enhanced the accuracy and reliability of microbial forensics, providing valuable tools for crime scene investigation and legal proceedings. This integration of microbiology into forensic science underscores the expanding potential of microbes in solving crime and administering the justice. Thank you so much. Thank you so much for inviting me. As time is very less, we have a constraint of time. So here I have shared overview of microbial forensics. If anyone has any question regarding this, so you may contact me in this number or you may directly mail. And because there are so many research scholars are working under my supervision, and I am guiding them in various field of forensic science, and microbial forensic is one of the field. So if anyone has any question, may ask from me in given context. Thank you so much.
Hello, ma'am.
Yes.
Yeah, it was very excellent information that you have shared with us.
Yes.
Being a basic microbiologist, I felt this is a very interesting topic to know that microbes have a lot of potential even in the field of forensic science.
Yes.
So I think I have a very basic or a preliminary question.
Yes,
You said microorganisms, we are using microorganisms as fingerprint.
yes,
In the sense that to find out the location of the crime
yes.
that is committed. So if in case the after committing that crime, if the dead body is transported to some other geographical area, how will microbes how will it help to identify in which location the crime was committed?
Yes, yes, it is very simple, and I have told in my lecture during my lecture. Uh, in shifting of the bodies, only I explained that in shifting of the body, if criminal want to misguide the investigator for shifting the body from one place to another place, so with the help of microbes, we can only identify the place of the crime. That is one of the way to identify the place of the crime. There are so many ways, and as I already told, this is used in corroborative evidence. Along with microbes, we have to study about other evidences also. So as I already told, microbes are specific in a specific place, and if body is shifted from one place to another, crime where crime is committed, that is known as primary crime scene, and where the body is shifted, that is known as secondary crime scene. And if the place is entirely different, so microbes is originated after 1 or 2 days only, and that microbes are from only that location where body is found. Okay. And
Yeah.
after 1 or 2 days is body is shifted, then we can say that body is shifted because the microbes which are identified from the body is not from this place, because from the in a specific place there are specific microbes, isn't it? So
Yeah.
here I already asked that. So in this way we can identify the geolocation, or identify the place of the crime.
Okay, thank you, ma'am. But my doubt was if the crime is done in some land, like some land, and the body is drowned. So
Yes,
then
yes.
it just gets washed off. All the microbes on the surface of the body will get washed off. In
As I
that case,
already
how will it
told,
help?
yeah, you are correct, ma'am. Just as I already told that that is corroborative evidence. If that type of situation will commit, then we will go through the another branch of forensic science. If each, and that is why I told that that is used as corroborative evidence. Okay. And
Mm.
if bodies was, so we will not find the microbes. When we find the microbes, then only in that condition, with the help of microbes, we can identify the place of the crime. If it is not, then we will use another method. And for criminal justice system is no, is always depend on the so many evidences, not any single evidence. If that was, then we can go through the second clue, like for me, maybe footwear, maybe identification of the person. Okay.
Mm.
Like some we can identify the location of the person also with the help of his or her feature, isn't it?
Mm.
That comes
Mm.
under forensic anthropology,
Mm.
isn't it? So
Correct.
if it is present, if microbes are present, then we can able to identify the place of the crime. If it is not, then we go through the other evidence.
Okay. Thank you so much, ma'am.
Yes, yes. And we will have, I will very happy if you will ask, all of you will contact me, and if you have any doubt, because in corona we can connect each other from various places, and that is the mode of connection of various people in the same platform. So we can exchange our ideas, we can exchange our thoughts. In this manner we can excellence in we can able to excellence in our education system also. Thank you. Is there any question?
Ma'am, can I ask one thing?
Yes, why not?
Uh, Ma'am, I want to know, when there are so much security, wherever there is a crime, we know that the area is blocked. But then too many crimes are not solved. Because of some powerful person has done it, because of
Mhm.
some reasons.
Hmm.
So how that person, which is that forensic person who is present there, how it manipulates the things, ma'am? I want to know that.
Manipulate the things. Okay.
हाँ, suppose
Yeah,
करिए
yeah,
कि
yeah, yeah.
एक
Very,
जगह
very
पे
good
crime
question.
हुआ है.
I appreciate your question. Uh, for forensic scientist, ethics should be compulsory. Okay. It is said that forensic investigation should be unbiased. If it is biased, so how? That is the question, na?
Yes,
How
ma'am.
they manipulate.
हाँ,
So
क्योंकि अगर कोई एरिया में कुछ प्रॉब्लम हुआ है, कहीं पे कुछ क्राइम हुआ है, और
yes.
सपोज कोई पावरफुल पर्सन है वहां पे,
Hmm.
तो हम सुनते हैं ना कि भाई हुआ तो है, लेकिन
Hmm.
उसका आज तक रिजल्ट नहीं मिला हमको.
Yes, मैं
तो
बताती
इसका
हूँ
मतलब
आपको.
ये है कि द पर्सन विच इज इनसाइड, है ना, ही मैनिपुलेट्स द थिंग्स ना.
Very good question. So there is a term which is known as chain of custody. Chain of custody should be maintained. For example, if you have collected a sample, if someone has changed a sample during the transportation, then how can we find the result? That is one situation, isn't it?
ओके. जी जी.
If we collect the sample and when we contaminated the sample, then how can we find the result? So these are some situation in which we cannot find the result and wrong result, or sometime we received the wrong sample also. So for maintaining, so it should be necessary. We have to maintain the chain of custody. Chain of custody. The person who collected the sample, they give the sample to the person who transport the sample. So each and every person signed on that document. If any sign is missing or any seal on the sample is breaked, then it may be possibility that chain of custody should may be breaked. Isn't it? So there are so many ways.
But this is one of the way, madam, that samples are carried from one place to another. But the place where the crime has taken place, as you have said, that fingerprints, many things
Yes, yes.
here, many things are available, no?
Yeah, you are correct. As I already told, as you all know very well, necessity is the mother of all inventions.
Mm-hmm.
Why we are not using a fingerprint right now? Because everybody knows we have to wear the gloves,
Uh-huh.
isn't it? So
Mm.
we have to choose another method of identification. Okay, so that is why other methods come into existence, because necessity is the mother of all invention. Nowadays what happened, some person make mold of fingerprints of other. Okay, and then that mold they can use for perform any crime. So what is the second evidence? Right now there is a very interesting fact that is known as knuckle print, which is present on the dorsal surface of our hand. This is palmar surface and this is dorsal surface, and this is also very unique in nature. So that is why we use different evidence for analyzing the things. And I think in India there is a time to boom the forensic science, as our Honorable Prime Minister has started so many laboratories and so many teaching institute, and hope in near future will give good result. I hope so.
Okay, ma'am. Thank you.
Yes. Thank you, everyone. If session is completed, then can I exit? Should I'm asking from the host?
Ma'am, very good session, ma'am.
Thank you so much. Thank you.
Dear participants, if you have any queries, kindly share with ma'am. We are in the end of program.
Or you may mail your queries. No issue at all. Okay, thank you so much.
Ma'am, vote of thanks, ma'am.
Yes.
Good evening to one and all. It's my privilege to propose a vote of thanks on this occasion. Gratitude is not only the greatest of virtues, but the parent of all others. As we express our gratitude, we must never forget that the highest appreciation is not to utter words, but to live by them. I would like to extend a vote of thanks to all our presenters, organizers, and attendees for making this webinar a great success. Your contributions and participation have made this event successful. I would like to thank our presenters for sharing their knowledge and expertise with us today. Dr. Swati Dubey Mishra for not only sparing their invaluable time for us to grace the occasion, but also for enlightening with us their commendable talk on the subject. Your participation and engagement have made this a truly interactive and collaborative experience. Dr. Swati Dubey Mishra is one of the greatest achievers. She is a record holder. She is the multi-talented personality. She is the woman of decorum, dignity, diplomacy, and also the most inspirational personality, ma'am. Thank you so much, ma'am. Finally, I'd like to thank all the participants. Thank you to everyone.
Thank you. Thank you. Thank you so much once again for inviting me. Thank you.